within those kind of weekly worksheets, what I'll end up doing is using those as a starting point. So I could easily see, rank the following sets, compounds, that says sets because there were multiple. There were multiple, there aren't in there right now. And I would just go through and change the structures. So, yes, following these same kind of patterns, I think there's a high probability. And thinking about all of that made me lose track of getting everything else. Organic name reactions. If we go back and look at our course content, not students, that's right. We officially cleared? No, we were doing this thing, huh?
Yeah.
So if we kind of talk through this again real quickly, when we look at these two reactions, ultimately the only thing that has changed is deciding which one acted as the acid or the base. So in this top case, that one acted as the acid, this one acted as the base. In the bottom case, we flipped it and said this was acid, and we said this was base. How would we know which one was correct? Okay, well, the example that we walked through in class is saying, okay, we just run it. We run them both, and we see what we get as products. And then what we would try and do is think through those products based off of our charge, size, electronegativity, resonance, induction, stability statements. The easiest structure to go through and identify would be our negatively charged species. Those negatively charged species show our electrons. Those are the easiest to manipulate because that's what we've spent the entire semester looking at is our electrons. And if we go through and compare those two negatives, well, they both have the same charge. They're both on the same atom. So that means the size of the atom that's holding the charge and the electronegativity of the atom holding the charge are identical. So there is no difference there. But the top case allows for resonance. That means that that negative charge is now spread across a larger area. Because it's spread across a larger area, what does that mean about its stability? It's more stable. What does that mean? This top reaction is a more likely outcome than the bottom reaction. Because it is a more likely reaction, what we can do is effectively ignore this one. Okay? So what we've done is took two reactions and just said, well, how do we think this would react? I don't know. Let's draw both reactions and see what happens and see if we can come up with a way to allow us to see that difference. Okay? Eventually what we'll do is start to pick up a little bit more familiarity with it. For instance, recognizing that we have a functional group called a carboxylic acid, which means more than likely it's going to act as an acid, and I should run the reaction according to that definition. Kind of make sense? Next part that kind of comes in, which is officially second semester general chemistry, is that when we look at this chemical reaction, yes, whoops, that's an eraser. Yes, it can progress in the forward reaction or forward direction, but it can also progress in the backwards direction. Okay? And what we're looking at is these two, it's maybe a single reaction, but it's two directions of that reaction that kind of compete with each other. And what we're talking about is the concept of equilibrium. So real quickly with equilibrium, if I had reactants and I had products, I'm going to build a trap door at the bottom of this container and I'm going to fill the reactants with liquid. Okay, so I've got a whole bunch of reactants. What happens if I remove the trap door?
The liquid goes into the product section.
The liquid moves over to the product section and we get a balance. Okay? It doesn't all move over to the product section, right? It eventually reaches the point where what's happening is the amount of particles moving over to the product side is equal to the amount of particles moving over to the reactant side. That's equilibrium. That's what's happening up here. Okay? It's our reaction going backwards and forwards. Okay? We can now further change this system a little bit. Okay? If we were to go through and look at this, we might go through and say, well, then all reactions are going to balance out so they get equal amounts on both sides. Well, that isn't necessarily the case. Why is it not the case? What happens if I change the size of the container? What if I make this a narrower container? There's more on the reactant side than there is on the product side. So all of our reactions have this kind of invisible balance of reactants and products with the invisible chambers behind them that we can't really see, but what we see is the end result. We can look at the amounts of products and compare that to the amounts of reactants. Okay? And we make that comparison, we can now start to quantify things. And what we end up quantifying this at, is something known as K, usually KEQ for equilibrium. Okay? And what it is the amount of products divided by the amount of reactants. That's what that means. So when we talk about our reactions, we could say a reaction had a K of 4. What does that mean?
Nothing if you don't have none of the reactants. Well,
I haven't specified how many products. I haven't specified how many reactants. K is just the ratio of products to reactants. If I have a value of 4, what does that mean about the ratio of products to reactants? There's more products than there are reactants. That's what it means. And if I had a K value of 0.4, what does that mean? I have more reactants than I have products. Okay? When we talk about these K values, we generally don't look at reactions that have these kind of relatively small K values. When we talk about our reactions, particularly for organic chemistry, the K values are usually like 10 to the 6th. Okay? That's massive. Why would we want a value that's like 10 to the 6th and not say 4? Why is 10 to the 6th a better K value than 4? Why do I want more products? It's kind of a stupid, obvious statement.
to see what happens during the
What's the point of organic chemistry? To make something. To make a lot of it or a little bit of it? A lot of it. The whole point is that I mix these reactants and they all go to products. That's the point of organic chemistry. If I run my reaction and I get a very small amount of products, that's not a useful reaction to run. So what I would do is change the reaction around to change that K value. Okay? Why do we talk about this K value? So next part is we're dealing with this scale. Okay? We get something up at that 10 to the 6th range. How many of you like powers of 10? Okay, we hate that. Okay? It's not easy to interpret that. It requires a little bit of time and patience to think through what that meant. So what scientists, for whatever reason, did was that, is there a way that I could change that numerical value, that power of 10, into a number that's easier for the human brain to compute, like 4? Okay? Could I do that? It turns out, yes, I can. How do I do that? I throw a bunch of fancy mathematics at it, and I take the negative log of that number. Now, that's just stupid. Why would we do that? If we take the negative log of both sides, what happens is we're no longer talking about K, we're talking about the negative log of K. We'll call that now a PK. Why? Why am I Mike? That's just the name we called it, PK. Okay? What happens over here? Well, it turns out the negative log of 10 cancels it out, and we get negative 6. The log of 10 cancels that power of 10, and it now turns it into a number that's a little bit more manageable for us to deal with. Okay? What does this mean for us? Well, that means instead of interpreting these large powers of 10, we now have to deal with interpreting smaller numbers, but we now have to deal with a p interpretation. Well, what is that p interpretation referencing? If I have a small pk, what does that mean? I had a large k. If I had a large k, what did that mean? Lots of products. So a small pk, lots of products. That's what all that means. Okay? And you can write all that mathematics down borderline utility. Okay? Kind of, sort of? Why is this super important for us? Well, most reactions, it isn't super important. Where it's important are the reactions we run a lot. What reactions do we run a lot? Acid-base. And so what we go through and do is we now say, instead of looking at all possible reactions, I say, for the reaction of an acid. Okay? So if I take HA, H plus A minus. If I now go through and look at that reaction, my products and my reactants change. I'm now looking at H plus. Those brackets are representing concentration over HA. Just changing my new expression around. Instead of this just being a general equilibrium expression now, it's the equilibrium expression very specifically for an acid, which means I don't call it KEQ anymore. I can call it KA. Yeah, you're like, this seems a little silly. Yeah. A, because it's now referencing an acid. I could now go through and look at the KAs for my acid. A large Ka means what? I have a lot of products. What is my product here? H plus. So a large Ka means lots of H plus, which means
Stronger acid.
there it is, a stronger acid. But our Ks were these weird powers of 10. I don't like powers of 10, so of course we have to say, oh, we do the conversion, and we convert it out to now a pKa. And I can now look at that number. So a small pKa means lots of H plus, which means stronger acid. Okay? Kind of, sort of? How could we use that? Well, we could go back to our equation here where we're like, well, how is this going to react? Okay, remember we said this one was the acid or this one was the acid. We didn't really know. Okay, and how did we figure it out? Well, we went through and we predicted based off all those charges and all those fun things. Which species should act like the acid? The stronger acid or the weaker acid? The stronger acid, that's why it's stronger. It should be the one that acts as an acid. We can look up the pKa of this species. The pKa of this functional group, or that structure, is about 4.2. Okay? You might say, well, that wasn't a negative number. That's true, but that's kind of a small number. So that's kind of acidic. Well, what's the pKa for this guy? I didn't expect you to know it. It's okay. 16. What does that mean?
Top one's strong.
The top one is the stronger acid. If I knew the pKa's, I didn't have to draw out structures to interpret structures. I could just say the top one's the acid, and the bottom one doesn't run. That species is now going to act as a base and react because it has a larger pKa. Kind of, sort of? So we can use that pKa information to predict which reaction is valid without having to draw a bunch of products. But that then also means that we've memorized a whole bunch of pKa values. Okay? Or we can look them up. Potato, potato, unfortunately. Okay? For me personally, looking at the charges is more relevant. Because memorizing the pKa works great to decide if the reaction is acting as an acid. Okay? But what if it's not an acid-base reaction? Well, now I can't use the pKa's anymore because the pKa's are only valid if it's acting as an acid. if I've memorized dealing with the structure stuff and how to interpret those, that simplifies later thought processes because it's now the same process. Look at the stability of the charges. Kind of, sort of? Okay. So there's all our things. And so the big kind of conclusion that comes out of this, those of you that may have to take biochem, I wouldn't be surprised if all of you take some level of biochem where you're looking at something like this. Okay? pKa. A large pKa value is a weak acid. A small pKa value is a strong acid. Well, what is a small value? It depends. Okay? This is all a relative scale, so we would actually have to know some numerical values to be able to make any utility of that. Forgetting that's not connected. These are some kind of ranges that you could see. Note the smallest one that we deal with is the carboxylic acid sitting about a 4, and the largest one gets up to our, where's our alkane? Our alkane sitting at 50. So when I look at the alkane functional group, I would expect it to not act as an acid because its pKa is large. So when we're saying small and large, small is close to 0, large is greater than 20. Okay? A good kind of center point that's useful to know would be our alcohols at 15. Not so much because of alcohols, but if we look at that functional group, it's an OH. Have we seen an OH functional group before outside of the context of an alcohol? Kind of like if we got rid of that carbon and we made that a hydrogen. Water effectively has a pKa about the same thing as an alcohol because they have, for the most part, the exact same structure. Okay? That's why their pKa's come in very similarly. Kind of makes sense? Knowing the pKa's can be helpful. It is not a requirement, but it does come in handy for some particular reactions. When they show up, I'll be like, yeah, remember when we said we should memorize our pKa's? Ta-da, this is where it would be useful. Okay? Generally what happens instead is people just memorize the reagents in those cases where the pKa would have been useful, and you just memorize the reagents and you're done. So it's deciding again, where do you want to spend the effort to memorize? Kind of, sort of? I did say we got a pKa down in the negative range. That's for our strong acids. Almost everything we deal with when we talk about acidity in organic chemistry is not strong acids. We use the strong acids to make all of our other functional groups react. Okay? Which gets me to another point that I want to talk about with our alkanes. We have a pKa to suggest that our alkane is not going to act as an acid. Could we have already predicted that that's not particularly acidic? Why?
No charge.
None of these other ones have charge.
It's happy.
Define happy.
It wants to stay where it is.
Why does it want to stay where it is?
Octets full.
All the other ones have octets full too. What you're doing isn't bad. You shouldn't give up. Like, that's good.
It just looks happy to me. Is it something with the forces?
Why would you say that?
Those are the gases.
Where are forces coming from?
Like the carbon to hydrogen bond.
And the carbon to hydrogen bond is a non-polar bond. Okay? Which means I wouldn't expect a large H+. It shouldn't act as an acid because that's a non-polar bond. It's equally sharing electrons. What happens with the carboxylic acid? That's a polar bond. And look at what happened to its pKa. Significantly more reactive. Alcohols, significantly more reactive. Amines, significantly more reactive. What's in common with all those significantly more reactive atoms? The bond type is all a polar covalent bond. We can predict that reactivity if we know the bond structure. That's why I push the bond structure so much, is we can come up with ballpark guesses that allow us to say, that bond is reactive, that bond is not. And knowing those connections allow us to predict something about that reactivity with very minimal knowledge. That's pretty powerful. Kind of, sort of?
That amine, a little less than 4, is that what you're saying?
No, the amine is 35 and 9, which gets a little...
35 and 9, I thought that was 35 over 9.
No.
Okay, there we go.
35
That's a little
and
less than
9. And amines become very tricky, okay? Particularly if you were going to move into biochemistry. Okay? Why do amines become tricky? Well, I see a hydrogen here. Okay? It turns out the pKa of that hydrogen is about 35, which is to say,
It's not
not very acidic. What's the 9 in reference to? The 9 is in reference to that lone pair could be donated to a hydrogen. So if we alter this structure and put a hydrogen there, our nitrogen becomes positively charged. That extra hydrogen now on there, that has a pKa of 9. Okay? So when you look up the pKa of an amine, what are they referencing? Are they referencing the pKa strictly of the amine, our red hydrogen, or are they referencing the pKa of the ammonium, the positively charged nitrogen, at 9? Almost universally, they're referencing the 9, which isn't for the amine, that's for ammonium. Okay? Why does this become important? Let's see if I can pull this off a little bit. We'll see how familiar you are with some of the future content that you might encounter in a different class. Let's just do this. What did I just draw above? Yeah. And it's fair. If you don't know, that's okay. No ideas? Okay. What is the name of the functional group now in blue? What is the name of the functional group in green? Carboxylic what? Acid. Guess what we drew? We have an amino acid. Okay? This is a relatively simple amino acid. This just has, or technically what we drew here is technically a protein, not an amino acid, because this has 2 amino acids within it. Okay? And so what you could do in a biochem class is acknowledge that that bond is the bond that connects the 2 different amino acids. Okay? Because the amine connects to the acid of the other one. Amine, acid. That is where our connection is. Kind of, sort of? Okay. Why has this become relevant when we're talking about pKa's? One of the most common things that we would do in a biochemistry class, or potentially, actually I'm not even going to guess, one of the things we could do in a biochem class is we would analyze proteins. Okay? To analyze those proteins, you have to separate them from other proteins. Well, how do we separate things? Okay? You're at kind of a loss on this. Reactions become a little bit dodgy to use to separate things, because when you react something, what happens to its identity? It changes, and it's no longer that compound anymore. So are you actually isolating the compound, or are you isolating the new thing? And that becomes this weird kind of Pandora's box thing to deal with. That said, we can chemically alter it as long as it's easily reversible. Like an acid-base reaction. Acid-base reactions are easily reversible. Okay? What would those acid-base reactions do? Well, they would either place hydrogens on the structure or remove hydrogens, depending on whether we were in acidic or basic conditions. So what we could go through and do with this amino acid is say, well, what happens to it? That's not the one I want to erase. I need that one. What happens to this amino acid if I'm at a pH equal to 1? Oof, what's a pH 1 mean? It's acidic. So I'm now saying I take this amino acid in an acidic environment. What happens to it? Okay, well, I have to know something about the chemistry of this molecule, okay, of each of those potential reactive sites to decide what would happen, if anything. If I'm acidic, what should happen to my amine? Well, the pK of the amine is 9 or 35. 35 is talking about removing the hydrogen that's already on there. I'm at a very acidic environment. Am I going to talk about removing that one? No, I'm acidic. Okay? I'm at a pH of 1. That's actually even lower than my pK of 9 for this one. What does that mean? This will act as a base, which means if I'm at a pH of 1, is this the correct drawing? No, it acted as a base. What does it do to act as a base? Define base. At a pH of 1, that nitrogen is positively charged. Okay? How about this group? Its pKa was 50. Okay? We're more acidic than that pKa, which means this should stay protonated. Does it have all of its hydrogens already? Yep, so nothing changes with it. How about here? This has a pKa of 10. I'm in an environment that is more acidic than that functional group, which means it should act as a base, and it needs to accept its proton. It already has its proton. How about here? That was the carboxylic acid, pKa 4. Already acidic, already has its proton. Why is this important to me? At a pH of 1, this molecule is positively charged. Can I take advantage of positives and negatives? Yeah, I could take if we think back to our silica plates, okay, and we can move through a different material. If you've heard of agarose, you can put an electrical field bias across the agarose, and that will separate things based on their charge. If I'm at a pH of 1, this is positively charged. What happens if I change the pH to a pH of 7? Well, is it more acidic or less acidic than my nitrogen?
Less.
It's still more acidic because it's a pH that is lower than my pKa, which means keep it protonated. What about the phenol? pH of 10, it's still lower, which means keep it protonated. What about the carboxylic acid? Now it's changed. For the carboxylic acid, the pKa is 4. I'm at a pH of 7. I am more basic than my carboxylic acid. What does that mean I should do? Remove the hydrogen. At a pH of 7, what is the charge on my protein now?
Neutral.
It's neutral, which means it's not going to separate according to the electrical field bias. That's going to change how it can separate. That's kind of cool. So if we're in a biochem class, what you're expected to be able to do is to deal with those pKa's interpretations of all of the amino acids in their side chains. And you should be able to predict the charge on an individual protein sequence. Okay? And that's usually where students flip out. But all you're dealing with is comparing the pH to the pKa. If the pH is lower than your pKa, make sure it's protonated. If it's higher, the pH is higher than your pKa, deprotonate it. That's it. But it does require that you know your protonated states and your deprotonated states. And where does that become particularly challenging? The amine functional group with that 9 versus 35. That 35 is too high for us to worry about. What's the max that we can deal with in biochem? It's a hard question. Give you a hint.
15.
Why 15?
Water.
In biochem, what's our solvent? Water. Which means any pKa larger than 15 is now more reactive than the water environment, which means we don't worry about it. So in biochem, because we're now limited in biological systems, the pH scale gets vastly narrowed and our pKa's become largely centered around that 15. Everything else we ignore, which is why we don't worry about the pKa of our CH3, because it is so high that to get to a spot where I'm going to do anything to that, I'm not going to be in water anymore. Well, if I'm in water anymore, not in water, I'm not with a biological system, and now I don't care because it's biochemistry. Kind of, sort of? Okay. So, how much of that do I expect you to be able to do? I think there's a practice question that's fair game in the weekly questions. I want to say aspartic acid, where you just look at a single amino acid and say, okay, at pH this, what should I expect it should look like? Okay? Kind of, sort of? Okay. After that, what we start moving into is now ultimately just kind of practice. Okay, we've got our 5 reactions. I fully expect you to be able to look at any reaction and be like, that's a substitution, that's an elimination, that's an addition. Which means you need to look at these and recognize what's happening in each of them. Some of the language you don't fully understand yet. Okay, LG, leaving group, which you understand because of lab. We talked about it in lab. We haven't fully addressed that in lecture. The details, the mechanism of how that happens has been addressed in lab. It hasn't been addressed in lecture, so I'm not going to ask on those details, but what I do expect you to be able to do is to look at the reaction and say, what happened? It was the exchange or transfer of an atom that was not hydrogen. I don't need to know what Lg is, but what I do need to note is that Lg is not in my product, or not in my organic product anymore. And it has been replaced by NuC. Should you know what NuC is?
Nucleophile.
That's nucleophile. And yes, you should know what that is. Okay? So when we're looking at these, what we're showing is all of the kind of constituent pieces that are associated with them. And what I want you to do is start looking for those big picture changes so that when you see a reaction, you can label it. So that if you think it's an addition, you need to see that pi bond disappear, and you need to know 2 sigma bonds have to appear. Because you may initially look at it and be like, I don't see that second sigma bond. Because that second sigma bond is almost always an implied hydrogen. And once you acknowledge that it's an addition, now you know I need to look for that hydrogen. But if you aren't acknowledging the reaction type, you don't know what you're missing. That's why I really want you to be familiar with those reaction types. And you might look at it and be like, what the heck's going on with the radical one? Fair question. Honestly, I don't know. I don't think I'll put that one on this exam. Let's go with that. Okay. I would also argue the radical isn't super relevant for you. Well, damn. It does have some relevance. We'll talk about it eventually, but we won't talk about it for this exam. Okay? The summaries are written in text, kind of explaining what's happening behind it. Note the language, we're looking at pi bonds and sigma bonds. We're referencing back to those bond characteristics that we talked about in the first unit to make these connections through here. Kind of, sort of? Okay. After that, it comes, sorry, did you want to keep copying? After that, that now moves into our mechanisms, which is the how these things happen. Okay? When we're talking about the how, there's really two categories that we move in. We have to break bonds and we have to make bonds. Okay? So all of the mechanisms that we will look for is how those bonds are potentially broken. Note, in addition to reactions, we lost a pi bond. That's going to be cleavage. Okay? Heterolysis is this weird one, is where we just break a bond. Just go, I don't like that bond, done. That's heterolysis. Okay? Bond formation. Well, I want to make a bond. There's a positive, a negative, and I put them together. Coordination. Okay? Elimination. The result of an elimination was the formation of a pi bond. Of course, that's in our bond formation category. So if we can categorize where those things are happening and overlay them with our reactions, we can start to make predictions about what's going on within our reactions and how and why they work. That is powerful because that can now put us in a system where we can look at reagents and be like, I've never seen these things before, but this is probably how it works. That's cool. That's what I would like you to get. So the same thing I expect out of a full year course, and they struggle there. So I fully expect you to struggle here too. Okay? But that's what I'm pushing you towards. Okay? There's also a system. Wow, that hurt. Where you are both breaking and forming the bonds at the exact same time. There are 3 mechanisms that allow for those simultaneous systems. Okay? The subsequent slides are now worked through in the play posit. Do you have specific questions about any of these? Should we try and talk through some of them in the broad strokes? Talk through them, or you had specific questions? Talk through them. Okay? What's happening in this first reaction?
Acid base.
Okay? Where are you seeing that acid base?
The O gives up its hydrogen to the nitrogen.
That hydrogen has changed locations. Okay? That would suggest it's an acid-base chemical reaction. Okay? If it's an acid-base chemical reaction, some species is acting as an acid, another species is acting as a base. Awesome. Which species is acting as the acid?
The O is the acid and the nitrogen.
This one?
I was talking about the left.
That's why I asked. The left. This is acting as our acid. The other species is acting as our base. Okay? Cool. How did the chemical reaction happen?
The hydrogen is transferred.
So I did this.
The electrons of the nitrogen are shared with the hydrogen.
And this is getting back to our definitions. When we're talking about our chemical reactions, it's the motion of electrons, not the motion of atoms. Okay? Motion of atoms, they're always moving around. They're always bumping into each other. That doesn't mean a chemical reaction happened. You've been in a busy rock concert, right? Concert or any place like that. Okay, and you've bumped into people, right? Gotten real close with people. Did you have a kid? Because, I mean, isn't that how kids, you bump into each other. Ta-da, kid. No, there's a couple other things that go along with that. Just because you're close doesn't mean a bond changed. Okay? The bond changed by showing the motion of the electrons. So we don't want to talk about the movement of atoms. We want to talk about the movement of electrons. Our base, the electrons on our base, shared with the hydrogen. That's what makes the bond between the nitrogen and the hydrogen. But if we did that, we would exceed the octet on the hydrogen. So what else do we have to do?
Electrons in the bond go back to the oxygen.
The electrons in the bond go to the oxygen. That accounts for that extra lone pair. Kind of, sort of? What we've now defined is that it's an acid-base chemical reaction, and we've defined the mechanism, how the electrons moved. Because it was for an acid-base reaction, we call this an acid-base mechanism. We formed the bond at the exact same instant we broke the bond. That was a simultaneous mechanistic step. How about the next one?
Substitution.
Okay, why substitution?
Atom other than a hydrogen was transferred.
Okay, what atoms were transferred?
The bromine and the iodine.
The bromine and the iodine have changed their locations. How did that happen?
Sharing of electrons.
What electrons were shared?
Give me a sec here.
And you can keep letting Devin dig, but you could also jump in for the rest of the class at any point to help her out.
Bromine
Donate electrons where? Let's take a look at that sodium bromine bond. I understand where you're going with that. And it's a common observation, and it's one I always forget about. What kind of bond do we have between the sodium and the bromine? Ionic. What does that mean happened with the electrons?
They were transferred.
It means they're not shared.
Yeah.
So if you're going to tell me this bromine is sharing electrons with the iodine, you're now saying that they're sharing. That's an ionic bond. They're not sharing. Okay? So what does that mean? The bromine may do something. I'll absolutely grant you that. But I should never show an arrow from the bromine going to the sodium. Even if I'm going to draw them next to each other, that's an ionic bond. By definition, there's no sharing of electrons.
The electrons in the bond between the bromine and the carbon go to the bromine,
Why? So rude.
break that bond.
I want to break that bond. Why should the electrons in that bond go to the bromine and not to the carbon?
Bromine's more electron.
Bromine is more electronegative than the carbon. I would expect those electrons to move that way. Fair answer. What's another answer?
Bromine's larger.
No. I mean, yes, but not relevant.
It wants to be negative.
It's not just that it wants to be negative. When we look at the answer, that's an ionic bond, which means it splits into sodium ion and bromide ion. For that bromide to be negative, it has to have 4 pairs of electrons. If we draw on our electrons appropriately, how many pairs of electrons are around that bromine? There's only 3, which means to get that 4th, has to take it from the bond. Don't get a choice. Okay? So the structures of those things can also lead us to that very same answer where you did a very chemical explanation. We could have just said, well, it has to be that way. Make sense? Okay, so now what happens?
The bromine's gone, the carbon wants.
This becomes an interesting thing. You're saying the bromine is gone. If the bromine is gone, would I see this answer? All you've told me is that the bromine is gone, which means the iodine's not there, and I have a positive charge on that carbon. Is that what it actually showed? No. So you can't tell me the bromine has gone and then something. It's not a then. It's simultaneous. So what is also happening as the bromine is leaving?
The iodine with the negative charge
The iodine with a negative charge is
comes in.
coming in, and if we had better drawn that, the iodine with a negative charge would have had 4 lone pairs with that negative charge, and that iodine is doing what?
Simultaneously going to the
How do I know it's simultaneous?
because of the
Both my arrows are shown in the same step. If I wanted to break that into multiple steps, I would show the formation of the carbocation. I didn't, which means all of those arrows had to happen in the same step. Okay? What mechanism is that? Well, it's not acid-base because it's not transfer of a hydrogen. It's some kind of substitution mechanism. Because it all happened at once, it's our simultaneous substitution mechanism, which we call SN2. S for substitution. N for nucleophile. When the substitution happened, what happened? Iodide, acting as a nucleophile, forced the substitution reaction. There's two molecules involved. Which seems kind of like a stupid statement. That's absolutely 100% true. Okay? What happens in the last one? Is it acid-base?
Oh, no,
No. Is it substitution?
no,
No. What is it?
elimination,
It's an elimination. Why do I know it's an elimination?
the formation of pi bonds.
Elimination is the only one that results in the formation of a pi bond. Since I see the double bond there, I now know that I've done an elimination. Awesome. Okay? How? This is a hard one. And you just gave me like a side look like it wasn't that hard.
Me?
Yeah.
Absolutely not. That was
Okay,
a lot
just
of work.
checking. That's what it looked like to me. I was like,
I
what?
was looking at you like
What do you see had to happen? We may not know what the symbols are necessarily. I can help a little bit.
LG left.
LG what?
It left.
How did it leave?
Carbon. It took the
The electrons in that bond went to the LG. Why do you think they went to the LG and not to the carbon?
because it's most likely more electronegative.
Why? Why? And even that, I mean, you're right, but you don't know any context of what a leaving group is. So it's not that you're wrong, but I don't think you know why it should be more electronegative. What evidence do you have written up here would suggest that LG should have taken the electrons?
The negative charge on the outside.
When we looked to the product side, it's negatively charged. It started neutral, it became negative. It gained those electrons, yes. Okay? Which means if it took those electrons, more than likely it was electronegative. Yes. Right? What else happened? Let's try it this way. Let's attempt to break it into steps. Okay, we know it's simultaneous, so we've only shown one thing here. Okay, but if we break it into steps, sometimes that can help for us. If we had the leaving group leave, that means that carbon would be positively charged, yes? To make the double bond, what do I need? Electrons. As positively charged, which means it doesn't have electrons. I've got to find the electrons from somewhere else. What? Okay. What is that lone pair going to do? Because I can't take the electrons and just go, whoop, give it to you. It doesn't work like that. I could share the lone pair from the base, and I could share, but why would it share with that carbon? That carbon's not charged. If anything, it would share with this carbon, but if it did that, what have I done?
You just replaced.
I did a substitution reaction. Absolutely right. The electrons on that base thing are important. The base thing? What's the definition of a base?
H plus acceptor.
An H plus acceptor. We look at the product, what did it do?
Accepted that.
It accepted an H. Where did that H come from?
One of the carbons.
One of the carbons. Do better. There are 3 carbons. What's that?
The positive carbon.
The positive carbon. So that red carbon we'll call positive, right? Call blue and green. How many hydrogens are shown connected to that red carbon?
None right now. But there should be two.
Fair, I asked it incorrectly. There's 2 hydrogens, which means in our cation there'd be 2 hydrogens. If you want to get rid of one of those hydrogens, how many hydrogens should show up in your product? There should only be 1 hydrogen. Is that a proper drawing for my carbon? Why not? It's missing its octet, which means it wouldn't be neutral, which means what else is there? An implied hydrogen, which means, is this mystical hydrogen connected to our base one of the red hydrogens?
No.
No, because it would mess with my octet cycle. Okay, so maybe it's the green hydrogen. There were 3 there, right? Satisfy the octet. Go to the product. There's still 3 there, which means it's not a green one. It's 2, and we look at our product. How many are there?
1.
There's only 1. Where did our hydrogen come from?
The blue.
Okay. But if it does that, we're now making that bond between our hydrogen and the base. Fantastic. But we would exceed the octet because officially we did nothing to that bond. So that bond is still there. That's problematic because we would exceed the octet. So what happens to the electrons in the bond between the hydrogen and the carbon? That would be this blue bond. I'll erase the other hydrogen. So I'll show it in black. What happened to the electrons in that blue bond? Everybody see the blue bond I'm talking about?
Yes.
That one right there? Where did those electrons go?
They have to confirm.
Okay. Well, I heard 2 answers, which is great. Okay. We heard they go to the carbon. If they go to the carbon, what would happen? Drawing our intermediate structure, we'd have the lone pair there and we'd have a negative. Fair. The other answer I heard was they go to the bond. What does that result in? The pi bond over here. Where did the electrons go? To the carbon or to the pi bond? They went to the pi bond. Why did we not say they go to the carbon? Why don't we show this as our product?
Because there's still electrons coming from
The base already did its thing.
the base. Oh, it did.
And wouldn't you be able to
What's the relationship between this charge structure and our final answer? What is the relationship between these 2? Not to give it away. What is the relationship between those 2 drawings? They're resonance structures, which means they are one and the same. Hey, look at that. I even spelled it right. Okay. Why should I draw this one with it being neutral versus that one being charged?
It's more correct.
It's more correct to show it neutral. This goes all the way back to our Lewis structure rules back in the first unit. We want to draw our structure, our best resonance structure that is neutral, not the one that is charged. Kind of, sort of? So cool. That's now all happening at once. We now get 3 curved arrows as opposed to 2. Okay? Those 3 curved arrows, when taken together with the fact that we're forming an elimination or forming a pi bond, is what's known as an E2. E for elimination. What do we say the 2 was? 2 molecules involved. 1, 2. See, it still holds. Why don't we call it acid-base 2 or AB2? Because it's acid-base. It's simple enough that we just address it as acid-base. Okay? Kind of, sort of? Questions about it? Okay. So when we look at our substitution, what this goes through and does is it addresses some of those kind of pieces. LG being leaving group, our nuke or base. We'll change that around between nuke and base depending on what type of chemical reaction is running. If we go through and compare, let's actually go backwards. Okay, what did the iodine act as? What did Br act as? It acted as a leaving group. What's the difference between these two reactions? Are they completely different? What's the difference between those two? Be obnoxious. Delete that. They're identical. Okay. So those two different reaction types use almost identical species. We've got a leaving group. We've got solvents. We've got a nucleophile, which does what? Donates electrons. What does the base do? Donates electrons. Okay? The only difference is really what they're donating electrons to. In one case, it donates it to a carbon, which forces a substitution. In the other case, it donates the electrons to a hydrogen, which forces the elimination. They have the same primary components, but how they act changes very, very subtly. And that's why you need to be careful when you go through and look at these things, because I can draw as a nucleophile OH minus. OH minus is a nucleophile. It will absolutely act as a nucleophile. What else will OH minus act as? A base. How do you know which it should be doing? Well, one thing is to look reactant to product to help try and use the clues of your reaction to help provide that context. Okay? Other things you can do, which becomes potentially more advanced, and we'll see if we get that advanced, is trying to trigger off of what's happening with our substrate, okay, that organic molecule with our leaving group on it, and see how that changes and manifests our reaction. Sometimes it makes it obvious, sometimes it doesn't. And obvious is massive air quotes for the record. Okay? So our nucleophile base, we're trying to differentiate. That's the group that shares electrons with our electron deficient atom, be it either the hydrogen or the positive. If we were looking at an SN2, it's sharing it with not hydrogen, causing our leaving group to leave, and we've now done that substitution. If we look at a different example for another SN2, what's happening? The nitrogen sharing electrons in, and we get a leaving group. This one looks a little bit different. Why does this reaction look a little bit different? It's a larger leaving group. Does the size of any of these really matter? No. Okay? So don't fall into the kind of false sense that leaving group needs to be small. It just needs to leave with the electrons. Okay? For it to leave as the electrons, it has to stabilize the negative charge. Okay? It can do that by being electronegative. It can do that by being large. It can do that by resonance. It can do that by induction. It could do that by flipping all of those options. Okay? So don't start saying it's just an atom. It's just a thing that needs to leave. Is it stable when it leaves? Try to avoid putting that kind of false perception of the size of the molecule on it. For our E2, same general principle, except now what's it doing? It's reacting with a hydrogen. When that hydrogen loses those electrons, they have to go somewhere, and it results in the loss of a leaving group. Okay, whoops. E2s, these are more E2s. This top one would be a nice counterexample as well. Look at our leaving group here. The leaving group is really big, right? But why would that want to leave? Why does the nitrogen want to leave? Why is that a good leaving group? Yeah, but bromine's electronegative too. Wouldn't the nitrogen be a better leaving group? Why could it be a better leaving group? Look at the charge. It started positive. What happens when it leaves? It's neutral. Whereas the bromine starts neutral and becomes Negative. So we could make a prediction that maybe the nitrogen's a better leaving group because it comes off uncharged as opposed to charged. We're using those rule sets to help us explain some of that chemistry. Okay? Once we move into our stepped, so yes?
I just had a quick question. So whether or not the leaving group ends up charged or not, ideally it'd not be charged, but they're both considered leaving group charged.
Yes. So the leaving group does not have to leave as a negative charge or end as a negative charge. It just has to stabilize the electrons somehow. And there can be scenarios where you get very large leaving groups. Okay? In fact, if you study glycolysis, there's a bond cleavage in glucose, 6 carbons, and you turn it into 2 3-carbon molecules. One of those 3-carbon molecules is a leaving group. And it is stabilized through resonance. That's how that bond cleavage works. Okay? Typically, we look and go, oh my God, it must be some complicated. It's just a leaving group. And it's just looking at the stability of what those atoms are that are causing the leaving to happen. The rest of the connectivity of that atom doesn't change. It's just there. This one is also an elimination, but this one looks a little bit weird. Why is this one a little bit weird? So thank you for telling me to go back because I skipped over it. Why does this one look weird from our other eliminations?
The nitrogen starts negative and the bromine.
I end with a triple bond. When we set elimination, does it say how many pi bonds? No, it just says the formation of a pi bond. That pi bond can form over the top of other pi bonds. That's perfectly acceptable. It's still an elimination. Okay? So don't get sidetracked by all the secondary stuff. I really don't care that there's a double bond there. As long as I make another one, I'm okay. Okay? In this case, here we're combining two things. Why would we want to combine two things? Well, what happened in this top mechanism? What did we do?
Transfer the electrons to the iodine by giving bond.
The electrons in the bond went to the iodine. Okay, which means mechanistically we did a heterolysis. We broke a bond. Did we do an acid base? No. Did we do a substitution? Elimination? Addition? We didn't do any of those because it's not a complete reaction. It's only a part of a reaction. When we look at our reaction cycles, they will do both bond breakage and formation. You need both to complete it. What we've looked at here is just the breakage. What now needs to happen? I need the formation. To do that formation, I have to bring in another reagent. I would have to bring in something else other than the negative iodine to come in and share with that positive. That would be the other half of my reaction. That could be my coordination. Okay? We look at the next system. Again, same idea. Bond breakage, heterolysis. Last one, what's happening here? Br minus comes in and attacks the positive. This is now a bond formation. Okay? The mechanism behind that bond formation is now a coordination. If I stepwise two things, let's take a look at just the top one. If I take the heterolysis up here and I follow it immediately with a coordination, that says coordination, now I could answer the question, what type of chemical reaction was this? Now it's a substitution chemical reaction. Is it an SN2 mechanism? What happened in an SN2? When? At the exact same instant. Is my bond formation and breakage happening at the exact same instant? No. Those are two separate mechanistic steps, which means it's not SN2. It's still a substitution. I still need a nucleophile to do the substitution. Okay? But it isn't two reactants. And this becomes the weird statement. Okay? The number of reactants are in the slow step. When we talked about SN2 and E2, how many steps were there? One, which meant that was by definition the slow step. Which of these two mechanism steps is the slowest step?
Breaking.
Why breaking?
I don't know, it just seems hard.
What is the energy difference between a carbocation and this? Which one's the better drawing? This one. Why? It's neutral. What does that mean about this one?
You have to work to get it into that non-neutral state.
It's higher in energy. Which one's better, this one or that one? The far product, which means, is that going to be an easy thing to do? Start at a high energy state and go to a low energy state. You all walked when you came in here. What did you then do?
Sit down.
You sat down. How many of you are getting up and walking around? Not very many. Why? That's a high energy state to be walking around. Okay, that high energy state you started at, and then what happens? You sat down. You drop to the lower energy state. That process is really easy. And if you're old like me, it's usually accompanied with okay? That's the process that's going on with it. It's a release of energy. What does that mean? This first step is the slow step. How many things are involved in this first step? One. So this is a substitution mechanism that involves a nucleophile, so it is SN, and in its slowest step, it involves how many species? One. So this whole thing, both of those mechanistic steps combined, can be referred to SN1. And that's where that name is coming from for those people in lab. Okay? Or you'll hear reference to in any other kind of discussion on this stuff. Okay? What it's necessarily going to introduce is this concept of carbocations. We already talked about carbocation stability. Tertiary is more stable than secondary, more stable than primary, more stable than zeroary. That conversation gets a bit misleading because what you immediately then assume is that tertiary is more stable. So here's two structures. Which carbocation is more stable? Red or blue? How do we classify? Tertiary, secondary, primary, zeroary. What do we do?
How many carbons?
How many carbons are connected to the red one?
3.
3, so we would call that tertiary. How many carbons connect to the blue one?
Wait, 2. Sorry, I thought you were talking about the other side.
Okay. 2, so we call that secondary. So which one's more stable? Red or blue?
Right.
Red, except the blue one's more stable. What? That's not fair, Mike. You can't do that. We're talking about substitution patterns to explain stability. How do we explain stability? Where do we start? You may have started to... How do we explain stability? Where do we start? Charge, they're both positive, there's no difference, right? Then what do we move to? Size, they're both carbon, no difference. Electronegativity, there's no difference. Resonance, did we talk about resonance at all when we did tertiary, secondary? No, because where is it? It's after induction. It's in the oh crap, I've got nowhere else to find a comparison that I'm going to look at substitution patterns. Let's look at resonance. Does the red one have resonance? Okay. If you're slow on that, that's okay. You need to practice. There's no resonance available for the red one. Is there resonance for the blue one? Yup. What does that mean? The blue one, according to our list, we hit resonance. The blue one has resonance. The red one does not, which makes the blue one more stable than the red one. Oh, but secondary, tertiary, primary didn't enter into it because there was a difference earlier in our reactivity tree. You have to address that reactivity tree. When we're talking about our stability of carbocations in this tertiary, secondary, primary, zeroary thing, all we're trying to do is be, what's the substitution difference? We have to realize that our molecules are vastly more complicated than that, and resonance and induction and other things can come into play before we get to the substitution patterns. Make sense? So when we think about our stability of carbocations, if you're insistent on trying to memorize a trend, it's okay to do this. And then what we can say is primary plus what? Resonance. Secondary plus tertiary plus. Okay? Nobody wants to memorize that as there's too many long things in there. Well, I agree, you shouldn't memorize that. Memorize charge, size, electronegativity. Deal with that. Okay? So if you're going to deal with stability of carbocations and you want to memorize something, okay, this is it. Tertiary plus resonance is more stable than secondary plus resonance, which is more stable than primary plus resonance, which is more stable than tertiary, which is more stable than secondary, more stable than primary, more stable than zeroary. Okay? There's one kind of gray area. The tertiary versus primary with resonance floats in a gray space. Okay? Depending on how you monitor that, you can see the tertiary being more stable, or you can see the primary with resonance being more stable. It's unfortunately a gray area. So you'll sometimes see those being roughly the same. Kind of, sort of? Okay. Oh, and I know this is about to get into I want you to be able to identify that a rearrangement has happened. I don't care that you note how it happens. Okay? So if we look at this, we're starting with a secondary carbocation. Secondary carbocations are not particularly stable. Okay? How unstable are they? The molecule will rearrange its atoms to make it more stable. So what happened is we took this bond and we moved that bond over there. This is kind of goofy. But if we do that, what happens? That carbon is now missing electrons. The carbocation is now at a tertiary spot as opposed to a secondary. Okay? Relatively minor. I don't think I'll test on rearrangements too much. That's probably a one question kind of thing. With that, I got to be done. And we'll pick up continuing to go through examples and that kind of fun stuff next week. And I will try, or not next week, today's Tuesday. On Thursday we'll do that.